Okay, so today we are going to be talking about Punnett Squares and test practice. And here's a Punnett Square. And there's some of Mendel's P's, so we've learned about those. And this is our objective for the day. So given a genotype of the two parents, the student will be able to determine the probability of genotypic and phenotypic combinations of an individual using a Punnett Square. So that's what we're learning today. Okay, so first we're going to start with a little review of what you should already know. So an allele is a different version or form of a gene. We receive one allele from each of our parents, and the combination of alleles received from our parents determine our genotype. And our genotype, if you remember, is the genetic makeup of an individual, while the phenotype is the physical characteristics of an individual. And the phenotype is determined by the genotype and also by the environment. Dominant allele is an allele that cancels the recessive allele, and it's represented by capital letters. And a recessive allele is expressed only when no dominant alleles are present. So recessive alleles are represented by lowercase letters. Homozygous. Homozygous is a pair of alleles that contain two of the same type of allele. So there's either two dominant alleles or two recessive alleles. So they're homozygous, the same type. While heterozygous is a pair of two different allele types. So for example, there's a little b and then a big B, so dominant and a recessive. Okay, so Punnett Square. A Punnett Square is a tool for predicting what offspring will look like when mating plants or animals. Here's some animals that have babies. And so the Punnett Square was invented after Mendel did his pea plant experiments by a man named Reginald Crundle Punnett. So that's why they're called Punnett Squares. So why do we study genetics? So we study genetics so we can predict the chances or the probability of inheriting or receiving certain traits. Here's some cats, a black and white cat. So have you ever wondered if a black and white cat breed, what their kittens will look like? What will their babies look like? So there's lots of different cat types. So it could be, is it going to be black, or it could be white, or maybe a mix of the two, gray? So how do we know what the kittens will look like? So those are what Punnett Squares are used for. They help with that. So I'm going to start off learning about the Punnett Square in general. So this is a Punnett Square. So it's basically just a square with a line down the middle and then a line down the center, and it makes four smaller squares. And the outside of the square is where the genetic contribution of the parents go. So the possible alleles that the parents can give to their children or their offspring. So that goes on the outside of the square. And then the inside of this, that goes on the outside of the square. So usually one parent goes on top. It's usually the father. So the alleles from the father go on top like this. And then the alleles from the mother go on the side. So one goes on the top and the other one goes on the left side. The inside of the Punnett Square contains the possible allele combinations of the offspring. So the parents are on the outside and then the offspring are in the inside. So possible offspring allele combinations. So parents can be homozygous, which have the same type of allele, or heterozygous, which is a different type of allele. Okay, so when we have the allele from the parents, They each have 2. We place them here. So how we use the Punnett square is we take the allele from the father that's here, and we put it in this box, and then we also put it in this box here, and then we take the allele from the mother and put it in this box, and then this allele from the mother goes in this box. So we'll fill in all the boxes using the alleles that come from the father and then from the mother, and then in this, for example, in this square we would use the allele from the father here, and then this is for the mother here. So we fill in these boxes here to find out what the offspring would look like. Okay, we're going to do a little example using cat color, like we talked about earlier. So we have the allele for the black cat color, or cat coat color, is a big B. And then the white coat of the cat is going to be represented by a little b, which is a recessive allele. So the big B is dominant over the little b, which is the white coat color. Or the black allele is represented by a big B because it's dominant. Okay, so if the dad has a black coat and he is heterozygous for coat color, he will have a big B and a little b. So he'll have one dominant allele and one recessive allele. And he will be black, have a black coat because he has a big B. So this is the dad. And the mom has a white coat. So because she has a white coat, she will have 2 little b's, or 2 recessive alleles, because she doesn't have any dominant alleles to make her coat black. So here's the mom. And here's our Punnett square. So first we place the dad's alleles on top here. So he has one big B and one little b. Next we place the mom's alleles on the side. So she has 2 little b's. And then we fill in our Punnett square. So our first square is going to look like this. So we're going to have one big B from our dad. We'll pull that down here. And then one little b from our mom. And then we fill in all the other 3 squares the same way. So the next one, we'll have a big B from our dad and a little b from our mom. And the next one, going to have a little b from our dad and a little b from our mom as well. And the next one is the same, 2 little b's. One from our dad and then a little b from our mom.